Today, I will talk about central tolerance in the immune system. Central tolerance is a mechanism that eliminates or functionally inactivates self-reactive T cells and B cells during their development. This process occurs in two main organs, the thymus for T cells and the bone marrow for B cells. The main purpose of central tolerance is to prevent the immune system from attacking the body's own tissues, which would otherwise lead to autoimmune diseases. In this slide, the diagram shows how central tolerance works and how it differs from peripheral tolerance, which acts later in the immune response. First, let's look at how central tolerance works in T cells. T cells development occurs in the thymus. During this process, T cells are tested for their ability to recognize antigens presented on self-MHC molecules. If a T cell reacts too strongly to self-antigens, it is considered dangerous because it may attack the body's own cells. These strongly self-reactive T cells are eliminated through a process called negative selection, which leads to apoptosis. This ensures that only safe T cells are released into the peripheral immune system. Here are the key points of T cell selection in the thymus. In the cortex of the thymus, positive selection occurs. T cells that weakly recognize self-MHC molecules receive survival signals and continue to develop. Then, in the medulla, negative selection takes place. T cells that strongly react to self-antigens are induced to undergo apoptosis. This two-step selection process ensures that mature T cells can recognize self-MHC but do not strongly react to self-antigens. Next, let's move on to B cell central tolerance. B cell development occurs in the bone marrow. Immature B cells are tested for their ability to bind self-antigens. If a B cell binds strongly to self-antigens, several outcomes are possible. The cell may undergo clonal deletion, meaning that it is eliminated. Alternatively, it may perform receptor editing, where the B cell receptor is rearranged to reduce self-reactivity. In some cases, the cell becomes anergic, meaning it is functionally inactivated. These mechanisms prevent self-reactive B cells from entering the circulation. Central tolerance plays a critical biological role in supporting autoreactive lymphocytes and protecting against autoimmune diseases. A key molecule involved in this process is AIR, which stands for autoimmune regulator. AIR allows thymic epithelial cells to express antigens that are normally found only in peripheral tissues such as pancreas or skin. This enables the developing T cells to be exposed to a wide range of self-antigens during negative selection. As a result, T cells that could react against peripheral tissues are eliminated before they become fully mature. In summary, central tolerance is an essential mechanism for maturing self-tolerance in the immune system. T cells undergo positive and negative selection in the thymus, which B cells are regulated through deletion, receptor editing, or energy in the bone marrow. Through these processes, central tolerance prevents autoimmunity and ensures immune system stability. Thank you for listening.